What is happening? Okay, sorry. So for this portion of the lesson, we're going to take a look at meiosis and genetic variation. Of course, meiosis is the process that creates gametes, sex cells, egg and sperm, and through sexual reproduction, combining those two gametes, we create genetic variation in populations, which is why no two individuals look identical. So let's start with genetic diversity. There's three contributing factors to genetic diversity. We've got fertilization, meiosis, and crossing over. So fertilization, of course, is when the sperm combines with the egg, and the thing is, it's totally random. It really just depends on which sperm gets to that egg first and fertilizes it. What this does is it increases the unique combinations of genes. So we see a shuffling, which again is why you don't look exactly like your parents or your brothers or your sisters. In humans, the chances of getting any one combination from a set of parents is 1 out of over 64 trillion different combinations. So you are incredibly unique. And it's also interesting to think about, what if it had been a different sperm that had fertilized your biological mother's egg? You could be sitting here with different hair color, eye color, possibly be the opposite sex. I know, it's totally random. Okay, next we have meiosis. We've been doing a deep dive into this process. Basically, meiosis creates the gametes. Something that happens that we're going to talk about a little bit later is the independent assortment of the chromosome pairs during metaphase. And what this independent assortment does is it also increases the unique combinations of genes, because the homologous chromosomes will pair up randomly at that metaphase plate or the cell equator. And they just randomly get distributed as meiosis continues. And so that creates about 8 million different combos of chromosomes that can result in the eggs or sperm. Then we have crossing over. Crossing over is kind of like what it sounds like. It's the exchange of chromosome segments between homologous chromosomes during prophase 1 of meiosis 1. So it only happens in prophase of meiosis 1. What this does is it creates new combinations of genes due to recombining chromosomes that are a combination of genes from both the mother and the father. So it creates sections of genes, sequences that didn't exist in your parents but do exist in you. And then you'll have a separate crossing over event for when you reproduce someday. So let's look at the mechanism of crossing over a little bit. So again, this happens in prophase 1 of meiosis 1. Remember, homologous chromosomes are basically, they're the same size, they carry the same genes, which I've indicated here with these little black bands that you should add to your handout. So what happens is, during synapsis, so that's that process, the two homologous pairs of chromosomes with the chromatid extensions here will physically wrap around each other, and that area where they're wrapping around is called a chiasma, and that's where the crossing over happens. And so during that crossing over event, we refer to those as a tetrad. So it's all 4 chromatids of those homologous chromosomes. So again, synapsis is the process, the chiasma is where the crossing over event is actually happening, and we call that a tetrad. And then you're going to draw the result of crossing over in the blank box there on your handout. So you can see I didn't do a great job. Basically, it's just kind of like puffy X's, and we just want to show that one chromosome now has sections from the darker chromosome, and then this one has sections from the lighter chromosome. Okay, so now we'll get into independent assortment a little bit. All right, now meiosis 1 is just the separation of the homologous chromosome pairs. So their job is to get the buddy chromosomes, if you will, away from one another. So all the homologous pair for pair 1, thinking about our karyotype, they're going to go into different sex cells. So will the 23rd pair, which will be the sex chromosomes. Those are also going to split. And so it's due to this independent assortment, which is where, like we mentioned earlier, these chromosomes are going to line up at the metaphase plate or the center in random ways. Now let me show you a color version so we can track this. So here is our body cell, right, that's getting ready to become, it's like the pre-sex cells. And we can track these with the color. So one way, and we're only showing 4 chromosomes to make it simple. It would be impossible to show all 46 of ours. So you can see one way, this cell gets the blues on the left, if you will, and the reds on the right, or they can go in this combination. And so if we follow here, then during meiosis 2, we're going to separate the chromosomes even further. And so you can see the idea is this result is we have 4 possible combinations here, or we can see that they shuffle and sort independently over here. So we get different results due to that independent assortment. So let's run down the left side. So like I said, meiosis 1, its job is to separate the homologous pairs. So meiosis 1, everything's getting ready. Then when we start meiosis 2, we have had homolog separation, which is why the chromosome with the dominant allele is here and the one with the recessive allele is here. Then meiosis 2 is going to be responsible for separating those sister chromatids so that each new sex cell only has half the amount of genetic information. So meiosis is going to reduce the amount of chromosomes, genetic information, to half so that when the egg and sperm combine, it can be restored. Now let's look at the haploid diploid. So over here, and it's the same diagram, it's just showing, the different results due to independent assortment. But during meiosis 1, we refer to those cells as having the diploid number of chromosomes because both of the homologous chromosomes are present. Here, now we've got separation of the chromatid copies. This is technically haploid because they only have one of the homologous chromosomes, but they still have their replicated copies. Then at the end, we would say those are totally haploid gametes and the replications have been separated. Okay, I hope you found that helpful. Next, you're going to go to the link for the video explaining the changes in DNA content during meiosis 1 and meiosis 2.